Approaches and methods in the study of behavior. Complete CSIR-NET JRF Life Sciences lecture. The study of animal behavior, or ethology, has evolved into a multidisciplinary science that integrates principles from biology, neuroscience, psychology, ecology, and evolution. To understand behavior, researchers utilize two broad approaches: the proximate mechanistic and developmental, and the ultimate evolutionary and functional. This division was formalized by Nico Tinbergen, who proposed 4 questions that define the study of behavior. 1. What are the mechanisms underlying the behavior causation? 2. How does the behavior develop in an organism? Ontogeny. 3. What is the function or adaptive value of the behavior? Survival value. And 4. How did the behavior evolve over generations? Phylogeny. These form the cornerstone of behavioral analysis in modern biology and are essential to understand for the CSIR-NET. Proximate approaches focus on how behavior occurs. This includes studying neural, hormonal, and genetic mechanisms. For example, hormonal control of mating behavior in birds or the role of neurotransmitters in aggression are examined under the proximate lens. Additionally, proximate studies investigate the ontogeny of behavior, how behavior develops during an individual's lifetime. This often includes the roles of learning, imprinting, and experience during critical developmental periods. On the other hand, ultimate approaches deal with why behavior exists. These examine how a particular behavioral trait increases an organism's reproductive fitness and how it may have evolved through natural or sexual selection. For instance, why do some bird species exhibit elaborate courtship displays? These behaviors likely evolved to attract mates and enhance reproductive success. Modern ethology employs various observational, experimental, and comparative methods to study behavior. Observational methods involve naturalistic study where animals are monitored in their natural habitats without manipulation. These methods yield ecological validity and are crucial for understanding spontaneous behaviors like foraging, mating, and social interaction. In contrast, experimental methods involve manipulating environmental or internal variables under controlled conditions to assess their effect on behavior. For example, altering light cycles to study circadian rhythm patterns in rodents is an experimental approach. Behavioral genetic methods such as selective breeding and knockout gene experiments are also common, especially in model organisms like Drosophila melanogaster and Mus musculus. Comparative methods involve comparing behaviors across different species to infer evolutionary relationships or selective pressures. These methods are particularly valuable in understanding the evolution of behaviors like altruism, communication, and tool use. Neuroethological approaches, which integrate behavior with neurobiology, use techniques like electrophysiology and brain lesion studies to understand the neural basis of behavior. Additionally, psychological methods, especially from behaviorism, use conditioning paradigms like classical and operant conditioning to study learning and memory. Pavlov's dog experiments, classical conditioning, and Skinner boxes, operant conditioning, remain foundational, and CSIR-NET has tested concepts from these extensively. Quantitative methods have become essential in modern behavioral research. These include the use of ethograms, detailed behavioral catalogs, time budgets, and statistical analysis like ANOVA or chi-square to validate behavioral patterns. Behavioral ecologists often use optimality models, game theory, and cost-benefit analysis to explain the adaptive value of behaviors like foraging or mating strategies. For example, optimal foraging theory oft predicts that animals will maximize energy gain per unit time, and its assumptions and violations are tested using both observational and experimental approaches. Furthermore, the rise of molecular tools, such as gene expression studies using qPCR or RNA-seq and brain imaging techniques like fMRI and optogenetics, have pushed behavioral research into a molecular and systems neuroscience era. These approaches provide mechanistic insight into complex behaviors like aggression, fear, decision-making, and social bonding. Understanding animal behavior also requires awareness of ethical considerations and animal welfare, especially in experimental paradigms. Ethical guidelines laid out by CPCSEA and international bodies must be strictly followed while designing behavioral experiments. The CSIR-NET exam occasionally tests awareness of these principles, especially in the context of vertebrate models. In summary, the study of behavior is approached through multiple lenses: proximate and ultimate questions, naturalistic and controlled studies, comparative and evolutionary insights, and increasingly, molecular and neurobiological methodologies. A CSIR-NET aspirant must master these foundational approaches and methods, understand how they are applied to real behavioral questions, and be able to critically assess the strength of inferences drawn from each method. Previous year questions: CSIR-NET Life Sciences, December 2015, Part B. Which of the following is an example of approximate explanation of behavior? A courtship dance increases mating success. Females perform courtship dance to attract females. C. The behavior is triggered by testosterone levels. These species performing dance evolve better survival traits. Answer C. Explanation: proximate explanations deal with immediate causes like hormones or neural mechanisms. June 2018, Part B. Which of the following is not a method used to study animal behavior? A. Ethogram. B. Observational study. C. Quantum dot imaging. D. Operant conditioning. Answer C. Explanation: quantum dot imaging is a nanotech method, not standard in behavior analysis. December 2013, Part C. A researcher finds that when the light cycle is altered, hamsters show changes in activity patterns. Which method of behavioral study is being used? A. Observational. B. Experimental. C. Comparative. D. Ontogenetic. Answer B. Explanation: this involves manipulating an external variable under controlled conditions. Experimental method. Would you like to continue with the next topic in behavioral ecology or move to another unit like molecular biology, physiology, or developmental biology? You said. Proximate causation. ChatGPT said. Excellent choice. Let's now explore the CSIR-NET JRF Life Sciences topic: proximate causation in animal behavior, a vital subtopic under ethology, frequently tested in both Part B and Part C of the exam. This lecture is structured from basic to advanced, in dense paragraph format, no tables, with maximum detail, and includes 2 to 3 CSIR-NET previous year questions with answers and years mentioned. Proximate causation in the study of behavior, CSIR-NET Life Sciences full lecture. In the scientific study of behavior, the concept of proximate causation refers to the immediate, mechanistic explanations for how behavior occurs. It addresses the physiological, genetic, neurological, and developmental processes that generate a specific behavioral output in an organism. This concept is distinct from ultimate causation, which focuses on why behavior exists in evolutionary terms. The framework for understanding proximate and ultimate causes was formally introduced by Nico Tinbergen, who categorized the study of behavior into 4 questions: 2 proximate mechanism and development, and 2 ultimate function and evolution. In CSIR-NET, questions often test one's ability to distinguish proximate causes from ultimate ones in behavioral scenarios. Proximate causation involves 2 primary subcomponents: mechanistic causation and ontogenetic developmental explanations. Mechanistic explanations refer to the neurobiological, hormonal, sensory, and genetic machinery that is activated in real time to produce a behavioral response. For example, in a male songbird, the activation of brain nuclei like the HVC, high vocal center, and RA, robust nucleus of the archopallium, in response to photoperiod changes leads to the onset of singing behavior during the breeding season. Hormones such as testosterone surge during this time, modulating the behavior further. In this context, the behavior of singing is the result of immediate internal physiological changes triggered by external stimuli, increased daylight, which is a classic case of proximate causation. The genetic basis of behavior also falls under proximate analysis. Genes influence behavior by encoding proteins that are involved in neurotransmission, hormonal synthesis, or receptor sensitivity. For instance, the phosphate gene in mice affects maternal care behavior. Knockout of this gene leads to defective nurturing behavior due to disruptions in sensory processing in the olfactory bulb and amygdala. Similarly, fruitless gene fru in Drosophila determines the male courtship ritual. Deletion or mutation of this gene can feminize male behavior. CSIR-NET has previously asked about gene behavior relationships, particularly those governing instinctive behaviors. The second subcategory under proximate causation is ontogeny, how behavior develops during an organism's lifespan. This includes both innate programs and learning mechanisms. For instance, imprinting in ducklings, as studied by Conrad Lorenz, is an innate learning process that occurs during a critical period, yet its underlying neural plasticity and timing are part of developmental proximate causation. Likewise, in rodents, social grooming behavior matures over time and depends on both genetic programming and early social interactions. In humans, language acquisition involves an intricate interplay between genetic predisposition, e.g., FOXP2 gene expression, and environmental learning, a classic ontogenetic mechanism. Sensory physiology plays a vital role in proximate causation, as behavioral responses often depend on the detection and processing of environmental stimuli. For example, in male moths, detection of female sex pheromones triggers orientation and flight toward the source, mediated by olfactory receptor neurons in specific glomeruli in the antennal lobe. In frogs, the auditory system is finely tuned to species-specific mating calls, and the activation of these sensory circuits leads to approach or mating behavior. These are clear mechanistic explanations for behavior and are tested frequently in CSIRNet questions involving model organisms. Additionally, proximate causation does not imply conscious decision-making. Rather, it focuses on the chain of biological events that lead to observable behavior. Even reflexes and fixed action patterns, such as egg rolling and geese or escape responses in fish, are explained through neural circuit activation and motor outputs. These behaviors, although appearing purposeful, are automatically triggered by key stimuli, known as sign stimuli or releasers, and are produced by innate releasing mechanisms (IRMs), all of which fall under proximate causation. To sum up, proximate causation provides insight into the how of behavior by exploring internal processes like hormones, neural activity, gene expression, and developmental pathways. Mastery of this concept is essential for CSIRNet aspirants, as it forms the basis for experimental designs, behavioral neuroscience questions, and gene function correlations that are often tested. It complements the understanding of ultimate causation, and together they enable a full understanding of why behaviors exist and how they operate. Previous year questions. CSIRNet Life Sciences, December 2015, Part B. Which of the following represents a proximate cause of behavior in an animal? A courtship display increases mating success. B. Behavior evolved due to natural selection. C. The behavior is due to elevated testosterone levels. D. The behavior helps in territory defense. Answer C. Explanation. Hormonal control is approximate. Mechanistic explanation. June 2017, Part C. Knockout of the fruitless gene in male Drosophila leads to lack of courtship behavior. This observation supports which type of behavioral cause? A. Ultimate cause. B. Proximate cause. C. Functional cause. D. Phylogenetic cause. Answer B. Explanation. Gene knockout affects immediate behavior, indicating a proximate genetic mechanism. December 2013, Part B. Which of the following best exemplifies a study focusing on proximate causation? Investigating whether behavior improves survival. B. Observing if the behavior is present in ancestral species. C. Measuring dopamine release during mating behavior. D. Comparing behavior across related species. Answer C. Explanation. Neurochemical analysis during behavior is a proximate level study. Would you like to now proceed to ultimate causation next to complete the concept pairing, or should we move on to another major topic from the syllabus such as behavioral ecology, neuroscience, or developmental biology? You said, ultimate causation. ChatGPT said. Here is your CSIRNet JRF Life Sciences lecture on ultimate causation, crafted at the highest level of quality, with maximum detail, structured in dense paragraph format, and note tables. It includes 2 to 3 CSIRNet previous year questions with answers in years. This is written specifically to serve as your only study material. Ultimate causation, CSIRNet Life Sciences full lecture. Ultimate causation in behavior refers to the evolutionary reasons why behavior exists. It addresses the adaptive significance or evolutionary advantage of a behavior, essentially answering the question, why has this behavior evolved and how does it contribute to an organism's fitness? The framework for understanding ultimate causation was formalized by Nico Tinbergen, whose 4 questions of ethology include 2 ultimate explanations: function, adaptive value, and evolutionary history phylogeny. Unlike proximate causation, which explores the immediate physiological, genetic, or neurological mechanisms of behavior, ultimate causation looks into long-term evolutionary benefits involving natural selection, inclusive fitness, and reproductive success. The functional explanation of behavior under ultimate causation focuses on how a particular behavior increases the survival or reproductive success of individuals. For example, in ground nesting birds, distraction displays like the broken wing display help to divert predators away from vulnerable chicks. This behavior increases offspring survival and, therefore, the inclusive fitness of the parent. The function here is clear: protecting genetic investment by misleading predators. For example, in ground nesting birds, distraction displays like the broken wing display help to divert predators away from vulnerable chicks. This behavior increases offspring survival and, therefore, the inclusive fitness of the parent. The function here is clear: protecting genetic investment by misleading predators. Such behaviors persist in populations because they confer selective advantage and are shaped by evolutionary pressures. The second aspect of ultimate causation is evolutionary history, which involves tracing how behavior has emerged through phylogenetic relationships, common ancestry, and divergent evolution. For example, courtship dances in birds of paradise are highly elaborate, but similar displays exist in related species, indicating that such behaviors may have evolved from a common ancestral trait. This evolutionary continuity highlights the historical pathway by which behaviors have been retained or modified in different lineages. Comparative behavioral studies across species are often designed to uncover such phylogenetic patterns. Ultimate causation is best appreciated through examples that show how behaviors enhance inclusive fitness. For instance, alarm calling in meerkats involves individual risk but benefits relatives, supporting kin selection theory. Although the caller may expose itself to danger, the survival of kin, who share many genes, ensures the propagation of those genetic traits. Similarly, behaviors such as reciprocal altruism in vampire bats, where individuals share blood meals with others, are explained through evolutionary game theory, as these strategies yield long-term mutual benefits and increase overall fitness. Behaviors can also evolve via sexual selection, a form of natural selection that acts on traits influencing mating success. Bright plumage, complex songs, or aggressive male-male combat behaviors are maintained in populations because they enhance access to mates, even at the cost of increased predation risk. For example, in peacocks, the extravagant tail does not help in survival but plays a role in female choice, directly affecting reproductive success. Such traits are explained by ultimate causation as they contribute to differential reproductive output, which is central to the theory of Darwinian fitness. Importantly, a behavior can exist without a current adaptive value if it is a vestigial trait, a byproduct of another adaptive trait, or retained due to phylogenetic inertia. In these cases, the evolutionary history still informs its ultimate explanation. This distinction is critical in avoiding the adaptationist fallacy, the incorrect assumption that all behaviors must be adaptive. In CSIRNet exams, questions often ask students to distinguish between proximate and ultimate explanations, identify adaptive significance, or evaluate evolutionary pathways of behavior. Candidates must be able to connect behavioral traits with fitness consequences and evolutionary logic rather than mechanistic pathways. Understanding ultimate causation provides insight into why natural selection has favored certain behaviors over evolutionary time, shaping the behavioral diversity we see across species. Previous year questions. CSIRNet Life Sciences, June 2016, Part B. Which of the following is an example of an ultimate cause of behavior? A testosterone level rise during mating season. B. Neural activation of song nuclei in birds. C. Singing by male birds increases chances of mating. D. Dopamine release in response to female presence. Answer C. Explanation. This is about reproductive success, an evolutionary explanation. Deck 2014, Part C. Alarm calls in ground squirrels alert others of a predator but increase the risk to the caller. Which theory explains this behavior under ultimate causation? A selfish gene theory. B. Kin selection theory. C. Neural imprinting theory. D. Fixed action pattern. Answer B. Explanation. Kin selection explains altruistic behaviors based on shared genetic interests. June 2013, Part B. Which of the following describes a behavior study from an ultimate causation perspective? A studying serotonin levels during aggression. B. Measuring gene expression during foraging. C. Understanding how foraging increases survival and reproduction. D. Identifying brain regions activated during song learning. Answer C. Explanation. Focuses on adaptive value, key aspect of ultimate causation. Would you like to continue to the next subtopic under behavior such as fixed action patterns, learning and conditioning, or behavioral ecology? I can also cover methods of behavioral study, model systems, or anything else from the CSIR net syllabus. Let me know what you'd like next. You said, altruism ChatGPT said. You said, altruism ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on altruism, written in maximum detail, paragraph format, with no tables, and designed to be your only study material for this topic. It includes 2 to 3 CSIR net previous year questions with answers in year mentioned. This note is JRF level, aligned with CSIR net exam patterns and past trends. Altruism, CSIR net Life Sciences full theory lecture. Altruism in animal behavior refers to an action performed by an individual that reduces its own fitness while increasing the fitness of another. This concept appears paradoxical under the traditional Darwinian framework of survival of the fittest, where traits that reduce individual survival or reproductive success are expected to be selected against. However, various evolutionary theories explain the adaptive significance of altruism when viewed through the lenses of inclusive fitness, kin selection, reciprocal altruism, and group selection. The foundation of understanding altruism in biology lies in inclusive fitness theory, proposed by W. D. Hamilton. Inclusive fitness is the sum of an individual's direct fitness, own offspring, and indirect fitness, helping related individuals reproduce. According to Hamilton's rule, altruism evolves when R, B, and GT, C, where R is the coefficient of relatedness between the actor and the recipient, B is the reproductive benefit to the recipient, and C is the cost to the actor. This principle explains why animals are often altruistic toward close relatives. For instance, Belding's ground squirrels give alarm calls to warn relatives of predators, even though it increases the caller's own risk. For instance, Belding's ground squirrels give alarm calls to warn relatives of predators, even though it increases the caller's own risk. Such behavior persists because the genes promoting altruism are also present in relatives and are thus propagated. This genetic explanation leads to the concept of kin selection, a type of natural selection that favors behaviors benefiting relatives. In eusocial insects such as honeybees, sterile worker bees sacrifice their own reproductive potential to help the queen reproduce. This extreme altruism is explained by their haplodiploid sex determination system, which results in workers being more related to their sisters R equals 0.75 than to their own offspring R equals 0.5. Therefore, helping the queen produce more sisters results in higher inclusive fitness. Another important mechanism explaining altruism is reciprocal altruism, introduced by Robert Trivers. This model applies to non-relatives and is based on the expectation of future return benefits. The behavior is favored by natural selection when the cost to the actor is less than the benefit to the recipient, and when interactions are repeated with opportunities for reciprocation. A classic example is seen in vampire bats, where individuals share blood meals with others who have failed to feed, on the assumption that the favor will be returned in the future. For reciprocal altruism to evolve, the following conditions must be met. Individuals must interact repeatedly, recognize each other, remember past interactions, and punish cheaters. Group selection, a more controversial explanation, suggests that altruism can evolve if it benefits the group, even at a cost to the individual. While classical Darwinism operates at the level of the individual, group selection posits that groups with altruists may outcompete groups with only selfish individuals. Although once dismissed, modern interpretations such as multi-level selection theory acknowledge that selection can act at both the individual and group levels under specific conditions. Some behaviors appear altruistic but are better interpreted through selfish gene theory. Richard Dawkins. According to this view, genes act to ensure their own propagation, even if the individual suffers. Apparent altruism is actually gene-centered selfishness. Altruistic behaviors are thus seen as strategies by which genes increase their chances of replication by influencing behavior that benefits genetically related individuals. Altruism is also shaped by ecological and social factors. In species with cooperative breeding, like meerkats or African wild dogs, non-breeding individuals help raise the offspring of dominant individuals. Ecological constraints such as territory scarcity or high predation risk can make independent breeding too risky, making helping behavior the best available strategy for increasing future reproductive chances. In humans, altruism is influenced by cultural evolution, social norms, and psychological factors, in addition to biological mechanisms. Acts like charity or volunteering may provide social prestige, reciprocal benefits, or even indirect genetic benefits when performed within kin groups. Though more complex than in animals, human altruism still aligns with evolutionary principles when considering long-term fitness consequences. Altruism remains a frequent area of questioning in CSIR net exams, particularly within the topics of behavioral ecology, kin selection, and evolutionary theory. Aspirants must understand both mechanistic theories, Hamilton's rule, Trivers' reciprocal altruism, and examples from natural populations, with emphasis on the adaptive value and evolutionary logic of altruistic behaviors. Previous year questions. CSIR net Life Sciences, June 2014, Part B. Altruistic behavior in eusocial insects is best explained by a reciprocal altruism. B group selection, C kin selection, D cultural transmission. Answer C. Explanation: the sterile worker bees help rear siblings due to high relatedness. December 2016, Part C. According to Hamilton's rule, R, B, and GT, C altruism will evolve if a cost to actor is greater than benefit to recipient. B relatedness is low and benefit is small. C product of relatedness and benefit exceeds cost. D benefit and cost are equal irrespective of relatedness. Answer C. Explanation: R, B, and GT, C is a condition for altruism to be selected. June 2015, Part C. In vampire bats, sharing of blood meals among non-relatives is an example of a kin selection. B parental investment. C reciprocal altruism. D altruism based on group selection. Answer C. Explanation: the behavior involves helping with expectation of future reciprocation. Let me know the next subtopic you'd like to cover. I can provide full CSIR net level lectures on fixed action patterns, learning and memory, foraging behavior, or any topic from the life sciences syllabus. You said. Evolution group selection. ChatGPT said. Here is your CSIR net JRF Life Sciences lecture on group selection, designed to be maximum detail, paragraph-based, no tables, and 100% exam-oriented. It includes 2 to 3 previous year questions with answers in years, and is written as your only study material to crack the CSIR net Life Sciences exam. Group selection. CSIR net JRF Life Sciences full theory lecture. Group selection is an evolutionary hypothesis proposing that natural selection can act at the level of groups, rather than exclusively on individuals or genes. Under this concept, traits that may be disadvantageous to individuals can still evolve if they provide a net benefit to the survival and reproduction of the group. This theory became controversial in the 1960s and 70s as it seemed to contradict the gene-centered view of evolution, especially as formalized by W. D. Hamilton, George C. Williams, and Richard Dawkins, who emphasized individual and kin selection as primary mechanisms. However, group selection has seen a resurgence through more nuanced frameworks such as multi-level selection theory. In classical Darwinian theory, natural selection operates by favoring traits that enhance an individual's reproductive success. Therefore, any altruistic behavior that reduces individual fitness is expected to be selected against unless it benefits kin via inclusive fitness or is reciprocated. However, group selection theory suggests that groups composed of more cooperative or altruistic individuals may have a higher collective fitness compared to groups of selfish individuals. If cooperative groups survive and reproduce more successfully than selfish groups, the cooperative trait may become common, even if it is individually costly. A classic theoretical example involves a population divided into multiple small groups, genes. Within each group, selfish individuals may reproduce faster than altruists. However, groups with more altruists may be more cohesive, resist extinction better, or migrate and colonize more effectively. In the long run, intergroup selection may favor altruism despite intergroup selection favoring selfishness. This creates a tension between within group and between group selection, the essence of multi-level selection theory. In practice, group selection is difficult to demonstrate in nature because within group selection often acts faster and more strongly than between group selection. Selfish mutants can quickly invade and outcompete altruists within a group, potentially collapsing group cohesion. However, some behaviors and traits, especially in eusocial insects, cooperative breeders, and humans, suggest that group-level benefits can shape evolutionary outcomes under specific conditions. For example, eusocial insect colonies, like ants or bees, operate almost like superorganisms, where sterile workers sacrifice individual fitness for the colony. Though this can be explained by kin selection, the colony-level integration also exemplifies group-level adaptation. Similarly, cooperative hunting in lions or alarm calling in primates can be seen as increasing the survival of the group, which may outcompete less cooperative groups. In human evolution, group selection has been invoked to explain the emergence of traits such as cooperation, morality, and social norms. Human groups that were more cooperative likely outcompeted others in warfare or resource acquisition. Cultural group selection is a modern extension, where cultural traits that favor group cohesion spread because of group-level advantages. It is important to differentiate naive group selection, now largely rejected, from multi-level selection, which is mathematically robust and accepted in modern evolutionary biology. Multi-level selection acknowledges that selection can act simultaneously at multiple hierarchical levels, genes, individuals, groups, even species, depending on the ecological and genetic structure of populations. In summary, while individual selection remains the primary force in evolution, group selection or multi-level selection is accepted as a valid evolutionary framework when group-level differences in survival and reproduction significantly influence gene frequencies. For the CSIR net exam, one should focus on the historical debate, mathematical principles, examples from natural populations, and the integration of group selection into the broader modern synthesis of evolutionary theory. Previous year questions. CSIR Net Life Sciences, December 2015, Part B. Group selection refers to natural selection acting at the level of individuals. B. Groups of individuals. C. Genes. D. Populations without group structure. Answer B. Explanation. Group selection acts on traits that benefit the group as a whole. June 2017, Part C. June 2017, Part C. In a population divided into multiple groups, altruists benefit the group but have lower individual fitness than selfish individuals. What mechanism can maintain altruism in the population? A. Kin selection. B. Intergroup selection. C. Group selection. D. Frequency-dependent selection. Answer C. Explanation. When altruistic groups outcompete selfish ones, group selection can maintain altruism. Mock Test 2023, CSIR website. Which statement best describes multi-level selection theory? A. Selection acts only at the level of genes. B. Selection acts only at the level of individuals. C. Selection can act simultaneously at the level of individuals and groups. D. Selection at the group level eliminates individual level selection. Answer C. Explanation. Multi-level selection acknowledges the simultaneous action of selection at different biological levels. Let me know the next topic you want, whether it's kin selection, reciprocal altruism, cultural evolution, or anything else from evolution or behavior. I'll give you another full, paragraph-based lecture designed solely to help you crack CSIR Net JRF Life Sciences. You said, kin selection, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on kin selection, written at JRF level depth, paragraph style, no tables, with maximum word limit coverage, based on previous year CSIR questions, and tailored to be your only coaching material for the exam. Kin selection, CSIR Net JRF Life Sciences full theory lecture. Kin selection is a form of natural selection where individuals increase their genetic fitness not only by producing their own offspring but also by helping relatives reproduce, because relatives share common genes. The fundamental idea is that genes promoting altruistic behaviors can spread in a population if those behaviors increase the reproductive success of kin who carry the same genes. This concept was formally introduced by W.D. Hamilton in the 1960s, and it revolutionized our understanding of social behavior in animals. At the heart of kin selection is Hamilton's rule, which provides the condition under which an altruistic trait will evolve. It is mathematically expressed as RB and GT, C, where R is the coefficient of relatedness between the actor and the recipient, B is the reproductive benefit gained by the recipient of the altruism, and C is the reproductive cost to the actor. If the benefit to the relative weighted by the degree of genetic relatedness outweighs the cost to the altruist, then the behavior can be favored by natural selection. The coefficient of relatedness R represents the probability that two individuals share a gene by common descent. For example, R equals 0.5 for siblings or parent offspring, R equals 0.25 for half siblings or grandparent-grandchild, and R equals 0.125 for first cousins. Kin selection explains the evolution of altruism among relatives, where direct fitness is sacrificed to increase indirect fitness. This combined measure of direct and indirect fitness is termed inclusive fitness, and natural selection acting to maximize this inclusive fitness is what underpins kin selection. The concept is particularly useful in explaining behaviors that appear maladaptive at the individual level but are adaptive when considered in the context of shared genes. A striking example is found in eusocial insects such as bees, ants, and wasps. In many of these species, sterile worker females forego reproduction to assist the queen in rearing more siblings. In Hymenoptera, sex determination is haplodiploid, where females are diploid and males are haploid. This results in sisters sharing 75% of their genes, which means a female worker can pass on more of her genes by helping her mother, the queen, raise sisters than by reproducing herself. This high relatedness is a key factor in the evolution of complex eusociality and caste systems in these insects. Kin selection also operates in vertebrates. In cooperative breeding birds like the Florida scrub jay, offspring often stay with their parents and help raise younger siblings instead of breeding on their own. This delay in reproduction is adaptive because the helpers increase the survival of close kin and may later inherit territory or breeding positions. In mammals like meerkats, cooperative rearing of young and sentinel behavior are often performed by individuals helping close kin, thereby increasing inclusive fitness. It's important to understand that kin selection does not imply conscious awareness of genetic relatedness. Instead, selection favors behavioral mechanisms such as imprinting, spatial proximity, or chemical cues that bias helping behavior toward kin. In humans, kin selection may underlie strong family bonds and caregiving behavior among close relatives, although culture and reciprocity add complexity. Kin selection differs from group selection in that it focuses on gene propagation via relatives rather than group-level traits. It also differs from reciprocal altruism, where individuals help others with the expectation of future help, typically among unrelated individuals. Kin selection can act independently or alongside these other mechanisms in the evolution of social behavior. In modern evolutionary biology, kin selection remains one of the most powerful and widely accepted explanations for altruism, cooperation, and social structure, especially in species with stable family units. Understanding kin selection is crucial for interpreting animal behavior patterns, mating systems, life history strategies, and the evolution of complex societies. Previous year CSIR Net questions on kin selection, December 2013, Part B. The concept of inclusive fitness is used to explain a selection at the species level. B. The evolution of altruistic behavior among relative. C. Mutation pressure as a driving force of evolution. D. Adaptive radiation in island habitats. Answer B. Explanation. Inclusive fitness includes both direct and indirect fitness, explaining altruism among kin. June 2015, Part C. A sterile female worker bee helps her mother, the queen, produce more daughters instead of reproducing herself. According to kin selection theory, which of the following best explains this behavior? A kin selection promotes behaviors that reduce inclusive fitness. Bee worker bees are more related to their sisters than their own offspring. C worker bees gain direct fitness through helping deselection acts only on groups and use social insects. Answer B. Explanation. In haploidiploid systems, sisters share 75% genes, more than mother offspring relatedness. December 2022, Part C. According to Hamilton's rule, an altruistic trait is expected to evolve if A, R, and GT, B, C. B, C, and GT, R, B, C, R, B, and GT, C, D, R, B, and LT, C. Answer, C. Explanation. Hamilton's rule. Altruism evolves if benefit to kin RB exceeds cost C. Let me know the next topic you want, such as reciprocal altruism, evolution of social behavior, biological clocks, or sensory ecology, and I'll prepare the same ultra-detailed JRF level lecture for you. You said. Reciprocal altruism. ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on reciprocal altruism, written in maximum detail, no table, paragraph-based format, tailored to the JRF level, including 2 to 3 previous year questions with answers in years. This is crafted to be your only study material for cracking the CSIR Net Life Sciences exam. Reciprocal altruism. CSIR Net JRF Life Sciences full theory lecture. Reciprocal altruism is a form of cooperation in which an organism acts in a manner that temporarily reduces its own fitness while increasing another's fitness, with the expectation that the favor will be returned in the future. This concept was introduced by Robert Trivers in 1971 and extended the theory of altruism beyond genetically related individuals, as explained by kin selection. It provides an evolutionary framework to understand altruistic behavior between non-relatives, which cannot be explained by shared genetic material alone. The key conditions that favor the evolution of reciprocal altruism include repeated interactions between the same individuals, recognition and memory of past interactions to detect cheaters, a benefit to the recipient that is greater than the cost to the actor, a mechanism of retaliation if cooperation is not reciprocated. In reciprocal altruism, the cost incurred by the altruist is offset by the future benefit of receiving help, assuming a system of mutual cooperation is maintained over time. This concept can be observed in various animal behaviors where the actor's investment is eventually compensated by returned active assistance, leading to long-term mutual benefit. One of the best-known examples comes from vampire bats, which share blood meals with roost mates that have failed to feed. The sharing bat incurs a short-term cost by regurgitating blood, but this act is typically directed towards individuals who have previously shared with them. Studies show that bats remember past donors and are more likely to help those that have helped them before, even among non-kin, a classic case of reciprocal altruism. Another well-studied system involves primates. In species such as chimpanzees and baboons, individuals engage in grooming, which not only improves hygiene but strengthens social bonds. Grooming is often reciprocated. An individual that grooms another is likely to receive grooming in return. Moreover, grooming can be exchanged for other favors such as support in fights, mating opportunities, or food sharing. These social exchanges, though not simultaneous, create a network of delayed but mutual benefits that enhance the overall fitness of individuals in the group. From an evolutionary standpoint, reciprocal altruism can be vulnerable to cheating, where an individual accepts the benefit but fails to reciprocate. However, natural selection favors strategies to minimize cheating, such as punishment, exclusion from future cooperation, or partner switching. This leads to the evolution of trust, fairness, and social rules, especially in cognitively advanced species. In humans, reciprocal altruism is deeply embedded in social behavior, morality, and cooperation, and is often formalized in cultural practices. We engage in acts like lending, gift-giving, and cooperative labor, expecting future reciprocation. Complex human societies have developed norms and legal systems to enforce and encourage reciprocal behavior, making it a cornerstone of social evolution. Reciprocal altruism is not driven by shared genes but by shared interest in future interactions. It has been modeled extensively using game theory, particularly the prisoner's dilemma, where the best strategy for long-term interaction is tit for tat, start with cooperation and then replicate the partner's previous move. These models show that cooperation can evolve and be stable, even in populations of unrelated individuals, under repeated interaction scenarios. In summary, reciprocal altruism provides an evolutionary explanation for non-kin cooperation, especially in social animals. It relies on memory, recognition, and long-term interactions, and is a key element in the evolution of complex social behavior. For CSIR Net, understanding reciprocal altruism in contrast with kin selection, group selection, and selfish behavior is essential. Previous year questions. CSIR Net Life Sciences, June 2013, Part B. Reciprocal altruism differs from kin selection in that it involves cooperation among unrelated individuals. B. It leads to inclusive fitness. C. It occurs only in social insects. D. It is based on genetic relatedness only. Answer. Explanation. Reciprocal altruism occurs between non-kin based on repeated interactions. December 2016, Part C. Which of the following conditions is necessary for the evolution of reciprocal altruism? A one-time interaction between individuals. B. High mutation rates in population. C. Long-term association and memory of past behavior. D. High relatedness among group members. Answer. C. Explanation. Memory and repeated interactions are essential for reciprocal altruism. Mock test CSIR, 2022. A vampire bat shares food with another that is not related, but has shared food before. This is an example of a kin selection. B. Reciprocal altruism. C. Group selection. D. Altruism based on inclusive fitness. Answer. B. Explanation. This is cooperation among non-kin, expected to be reciprocated in future. Let me know if you want the next lecture on biological clocks, sensory ecology, evolution of social behavior, or any other CSIR topic. I'll give you a full JRF level, one stop note again. You said. Neural basis of learning, ChatGPT said. Here is your CSIR Net JRF Life Sciences lecture on neural basis of learning, prepared at JRF level, maximum word limit, paragraph based, with no tables, and includes 2 to 3 previous year CSIR questions with answers in years. This is designed to serve as your complete and only coaching material for this topic. Neural basis of learning, CSIR Net JRF Life Sciences full theory lecture. Learning is the process through which experience leads to a relatively permanent change in behavior or the potential for behavior. The neural basis of learning refers to the physiological and structural changes that occur in the nervous system, particularly the brain, during the acquisition and storage of new information. At the core of learning lies synaptic modifications, neurochemical signaling, and changes in neuronal circuitry. One of the most fundamental principles underlying learning is synaptic plasticity, the ability of synapses to strengthen or weaken over time in response to increases or decreases in their activity. This plasticity enables the nervous system to encode experiences, form memories, and modify behavior accordingly. 2 primary forms of synaptic plasticity that are critical to learning are long-term potentiation (LTP) and long-term depression (limited). Long-term potentiation (LTP) is a long-lasting enhancement in signal transmission between 2 neurons. Learning is the process through which experience leads to a relatively permanent change in behavior or the potential for behavior. The neural basis of learning refers to the physiological and structural changes that occur in the nervous system, particularly the brain, during the acquisition and storage of new information. At the core of learning lies synaptic modifications, neurochemical signaling, and changes in neuronal circuitry. One of the most fundamental principles underlying learning is synaptic plasticity, the ability of synapses to strengthen or weaken over time in response to increases or decreases in their activity. This plasticity enables the nervous system to encode experiences, form memories, and modify behavior accordingly. 2 primary forms of synaptic plasticity that are critical to learning are long-term potentiation (LTP) and long-term depression (limited). Long-term potentiation (LTP) is a long-lasting enhancement in signal transmission between 2 neurons that results from their simultaneous stimulation. It is considered a key cellular mechanism underlying learning and memory, especially in the hippocampus, which is involved in spatial and declarative memory. LTP typically occurs at glutamatergic synapses and is mediated by NMDA and methyl D-aspartate receptors, which are both voltage-gated and ligand-gated. When presynaptic glutamate release coincides with postsynaptic depolarization, NMDA receptors allow calcium influx, initiating intracellular signaling cascades that strengthen synaptic connections, either by increasing receptor density or altering gene expression. In contrast, long-term depression limited involves a long-lasting decrease in synaptic strength. It also occurs at glutamatergic synapses but often involves a low-frequency stimulation that results in a modest but sustained calcium influx. This activates different pathways, e.g., phosphatases instead of kinases, leading to synaptic weakening. The balance between LTP and limited is essential for synaptic scaling, memory formation, and forgetting, thereby refining learned responses. Another key player in the neural basis of learning is heavy plasticity, often summarized by the phrase, cells that fire together, wire together. According to this principle, if a presynaptic neuron repeatedly activates a postsynaptic neuron, the synaptic connection between them strengthens. Hebbian learning provides the framework for associative learning, including classical conditioning and operant conditioning. In non-associative learning such as habituation, decreased response to a repeated stimulus, and sensitization, increased response after a strong or noxious stimulus, specific changes at the neuronal level have also been documented. These forms of learning have been extensively studied in simple systems like aplesia, a C-slug used in neurobiology. In aplesia, habituation involves reduced neurotransmitter release at synapses, while sensitization involves enhanced neurotransmitter release due to modulatory interneurons and the involvement of second messengers like cyclic AMP. Neurotransmitters and neuromodulators are critical in learning mechanisms. In aplesia, habituation involves reduced neurotransmitter release at synapses, while sensitization involves enhanced neurotransmitter release due to modulatory interneurons and the involvement of second messengers like cyclic AMP. Neurotransmitters and neuromodulators are critical in learning mechanisms. For instance, dopamine is essential in reward-based learning and reinforcement, particularly via its action in the mesolimbic pathway, which includes the ventral tegmental area (VTA) and the nucleus accumbens. The role of dopamine in learning is not just about pleasure but also about prediction error, a mismatch between expected and actual outcomes, which updates future behavior. Brain regions involved in learning include the hippocampus, declarative and spatial memory; the amygdala, emotional learning, fear conditioning; the cerebellum, motor learning and classical conditioning; the prefrontal cortex, working memory, decision making. Brain regions involved in learning include the hippocampus, declarative and spatial memory; the amygdala, emotional learning, fear conditioning; the cerebellum, motor learning and classical conditioning; the prefrontal cortex, working memory, decision making, and extinction of learned behaviors; and the basal ganglia, procedural learning and habit formation. Molecular mechanisms supporting learning include gene expression and protein synthesis, particularly during the consolidation phase. Transcription factors like CREB, camp response element binding protein, are crucial for converting short-term plastic changes into long-term memory. Blocking protein synthesis often prevents long-term memory formation, demonstrating the link between molecular processes and behavioral learning. Learning also modifies the structural architecture of the brain. Studies using imaging and microscopy have revealed synaptogenesis, dendritic branching, and even adult neurogenesis in areas like the hippocampus, all associated with learning. In conclusion, the neural basis of learning involves a dynamic interplay of electrical activity, molecular signaling, synaptic remodeling, and structural changes in the brain. Understanding this complexity is essential to the study of behavioral neuroscience, neurodevelopment, and neurodegenerative disorders that impair learning and memory, such as Alzheimer's disease. Previous year CSIR-NET questions on neural basis of learning, June 2014, Part B. Long-term potentiation (LTP) in the hippocampus is associated with a decrease in synaptic strength. B. Increase in postsynaptic receptor density. C. Blockade of calcium influx. D. Decreased neurotransmitter release. Answer B. Explanation: LTP involves insertion of AMPA receptors into the postsynaptic membrane, increasing synaptic strength. December 2015, Part C. In an experiment on aplesia, repeated gentle stimulation of the cyclin results in reduced skill withdrawal. This is an example of a classical conditioning. B. Habituation. C. Sensitization. D. Extinction. Answer B. Explanation: aplesia shows reduced response to repeated, benign stimulus, a hallmark of habituation. June 2019, Part B. A critical component of synaptic plasticity underlying learning is a death of neurons in the hippocampus. B. Static synaptic strength over time. C. Activity-dependent changes in synaptic strength. D. Inhibition of neurotransmitter synthesis. Answer C. Explanation: learning requires activity-dependent modifications of synapses, such as LTP Unlimited. Let me know if you'd like the next topic: biological clocks, molecular basis of memory, evolution of nervous system, or any other CSIR life sciences topic. I'll prepare it with the same quality. You said memory. ChatGPT said. Here is your CSIR-NET JRF Life Sciences lecture on memory, written at the highest possible quality and depth, suitable for JRF level preparation. It is crafted entirely in paragraph form, with no tables, includes 2 to 3 previous year CSIR-NET questions with answers in year mentioned, and is designed to serve as your only study material for cracking the exam. Memory, complete theory for CSIR-NET JRF Life Sciences. Memory is the fundamental process by which organisms encode, store, and retrieve information. It is a vital function of the nervous system that allows an individual to adapt, learn from past experiences, and make decisions. At its core, memory is a consequence of neuroplasticity, whereby synapses, neuronal networks, and entire brain structures undergo biochemical, electrical, and morphological changes in response to experience. Memory can be categorized into various types based on time scale, consciousness, and neural circuits involved, including sensory memory, short-term memory, working memory, and long-term memory, which can be further divided into explicit declarative and implicit non-declarative forms. Sensory memory is the most immediate form of memory, lasting less than a second. It temporarily retains sensory impressions, like visual, iconic, and auditory echoic information, and serves as the initial filter before the information is processed further. Sensory memory depends largely on activity in the primary sensory cortices. Short-term memory (STM) retains limited information for a brief duration, usually seconds to minutes. The classic example is remembering a phone number long enough to dial it. Working memory is a refined component of STM that not only stores but also manipulates information in real time, such as solving a math problem mentally. Working memory is highly dependent on the prefrontal cortex, where executive control, attention regulation, and integration of multiple cognitive processes occur. Long-term memory (LTM) refers to the enduring storage of information for days, years, or a lifetime. It involves 2 major subtypes: explicit memory, which requires conscious recall, and implicit memory, which operates unconsciously. Explicit memory includes episodic memory, personal experiences, and semantic memory, facts and knowledge. These types are primarily associated with the hippocampus, medial temporal lobe, and neocortex. The hippocampus is critical for the consolidation of short-term episodic memories into long-term storage. Lesions in this area, as seen in the famous case of Henry Molaison (HM), result in profound anterograde amnesia, loss of the ability to form new long-term memories. Implicit memory includes skills and habits, procedural memory, classical conditioning, and priming. It relies on brain areas like the basal ganglia, cerebellum, and amygdala. Procedural memory enables learning of motor sequences, e.g., riding a bicycle, and does not require conscious recall. At the cellular level, memory formation involves changes in synaptic strength via processes like long-term potentiation (LTP) and long-term depression (LTD), particularly in the hippocampus. LTP, triggered by high-frequency stimulation, leads to increased calcium influx through NMDA receptors, activation of CAMP and PKC, and subsequent insertion of AMPA receptors into the postsynaptic membrane, resulting in stronger synaptic responses. Over time, LTP induces gene expression changes and protein synthesis necessary for stabilizing synaptic modifications, a process called memory consolidation. Transcription factors like CREB, camp response element binding protein, are essential for initiating these gene expression cascades. Blocking protein synthesis immediately after learning impairs long-term memory but not short-term memory, reinforcing the molecular distinction between memory stages. In addition to the hippocampus, sleep plays a major role in memory consolidation, especially for declarative memories. During slow-wave sleep, hippocampus-dependent memories are repeatedly reactivated and redistributed to cortical sites for long-term storage. This supports the 2-stage model of memory, where hippocampal networks initially encode information, which is later transferred to the neocortex for permanent retention. Forgetting, often misunderstood as memory failure, can be an active process necessary to prevent cognitive overload and optimize decision-making. Forgetting involves mechanisms like synaptic pruning, limited, and inhibitory regulation of memory retrieval circuits. The prefrontal cortex modulates the recall and suppression of memories, balancing memory retention with adaptability. Neurodegenerative diseases, such as Alzheimer's disease, are characterized by severe memory impairments. Alzheimer's typically begins with loss of episodic memory, progressing to semantic and procedural deficits. The pathological hallmarks include amyloid beta plaques, neurofibrillary tangles, and hippocampal atrophy, all of which disrupt the neural circuits essential for memory. In summary, memory is a multi-layered process involving encoding, storage, and retrieval of information through complex and interdependent neural, cellular, and molecular mechanisms. From synaptic plasticity to large-scale neural networks, memory illustrates how behavior, experience, and biology are intimately connected. Understanding its neural basis is not only central to neuroscience but also critical for addressing cognitive disorders, optimizing learning strategies, and developing therapeutic interventions. Previous year CSIR Net questions on memory, CSIR Net June 2018, Part C. A student performs a test in the morning and can recall the answers in the afternoon. Which of the following is most likely involved in the consolidation of memory during the day? A. NMDA receptor activation. B. GABA receptor upregulation. C. CREB inhibition. D. Inhibition of calcium influx. Answer. A. Explanation. NMDA. Previous year CSIR Net questions on memory, CSIR Net June 2018, Part C. A student performs a test in the morning and can recall the answers in the afternoon. Which of the following is most likely involved in the consolidation of memory during the day? A. NMDA receptor activation. B. GABA receptor upregulation. C. CREB inhibition. D. Inhibition of calcium influx. Answer. A. Explanation. NMDA receptor activation is essential for long-term potentiation and memory consolidation. CSIR Net Deck 2015, Part C. Blocking protein synthesis shortly after learning a task affects A. Only short-term memory. B. Only long-term memory. C. Both short and long-term memory. D. No effect on memory formation. Answer. B. Explanation. Long-term memory requires new protein synthesis, unlike short-term memory. Explanation. Long-term memory requires new protein synthesis, unlike short-term memory. CSIR Net June 2021, Part B. The hippocampus is primarily involved in which type of memory? A. Procedural memory. B. Classical conditioning. C. Declarative memory. D. Priming memory. Answer. C. Explanation. Declarative memory, episodic and semantic, relies on the hippocampus for encoding and consolidation. Would you like the next lecture on biological clocks, sleep and memory, learning and memory integration, or neural circuits in behavior? Let me know, and I'll prepare it just as thoroughly. You said, cognition, ChatGPT said. Here is your CSIR Net JRF Life Sciences full-length lecture on cognition, written at maximum detail, entirely in paragraph format, with no tables, and tailored to the JRF level based on previous year question trends. It includes 2 to 3 solved previous CSIR Net questions with year-end answers, and is optimized to serve as your exclusive, all-in-one coaching material for exam success. Cognition, complete CSIR Net JRF Life Sciences lecture. Cognition refers to the mental processes and faculties involved in acquiring, processing, storing, and using information. It encompasses a wide array of high-level brain functions, including perception, attention, learning, memory, language, reasoning, problem-solving, and decision-making. In biological terms, cognition is not simply abstract thought. It is deeply rooted in neural architecture and shaped by evolution, experience, and environment. The cognitive capabilities of an organism directly impact its survival, reproduction, and social dynamics, making cognition a central theme in behavioral neuroscience, evolutionary biology, and psychology. At the neural level, cognition arises from coordinated activity within and across different regions of the central nervous system, particularly the cerebral cortex, which houses areas such as the prefrontal cortex, temporal lobe, and parietal cortex, all of which play specific roles in cognitive operations. The prefrontal cortex (PFC) is the executive hub of cognition, responsible for attention regulation, planning, impulse control, working memory, decision-making, and social behavior. Lesions or dysfunctions in the PFC often result in cognitive deficits such as poor judgment, impulsivity, or inability to plan future actions, as seen in frontal lobe syndrome and various psychiatric disorders. Perception is the first cognitive step, involving the interpretation of sensory input from the environment. The primary sensory cortices receive inputs e.g., visual, auditory, tactile, which are then integrated in association areas to generate a meaningful representation of the environment. From this base, cognitive functions such as attention selectively enhance relevant stimuli while suppressing distractions, a process mediated by dopaminergic and noradrenergic neuromodulation in the PFC and parietal cortex. Learning and memory, as already discussed, serve as cognitive foundations for the storage and retrieval of past experiences. Working memory, the transient holding of information for manipulation, is a crucial component of cognition and is tightly regulated by prefrontal circuits in coordination with parietal and hippocampal regions. Importantly, cognitive flexibility, or the ability to adapt thinking and behavior in response to changing environments or rules, depends on the dynamic interplay between the prefrontal cortex and subcortical structures such as the basal ganglia. Language, one of the most sophisticated cognitive functions, involves specialized brain regions in humans, Broca's area for speech production and Wernicke's area for language comprehension. While complex language is unique to humans, other animals exhibit forms of symbolic communication and vocal learning. While complex language is unique to humans, other animals exhibit forms of symbolic communication and vocal learning, offering comparative insight into the evolution of cognitive processes. In birds, for example, song learning in zebra finches requires dedicated neural circuits like the high vocal center (HVC) and RA nucleus, which are shaped by both genetics and experience. Decision-making and problem-solving represent the most advanced levels of cognition. They require evaluating alternatives, anticipating outcomes, and choosing optimal strategies. These processes are influenced by both rational computation and emotional modulation, the latter involving structures such as the amygdala and orbitofrontal cortex. Neuroeconomic studies reveal that even non-human primates and rodents exhibit rudimentary forms of decision-making, including risk assessment and delayed gratification, illustrating that the roots of cognitive processes extend deep into the animal kingdom. Social cognition, the capacity to perceive, interpret, and respond to the behaviors and intentions of others, is crucial in animals that live in complex societies. It involves theory of mind, empathy, and recognition of social hierarchies. Primates, elephants, dolphins, and certain birds such as corvids show impressive cognitive abilities such as near self-recognition, imitation, and tool use. These behaviors are interpreted as evidence of higher-order cognition, supported by evolutionarily expanded cortical regions and enriched life experiences. On the molecular and cellular level, cognition is underpinned by synaptic plasticity, neurotransmitter dynamics, and gene regulation. Glutamatergic signaling, especially via NMDA receptors, is central to synaptic changes involved in learning and problem-solving. Modulators like dopamine and serotonin influence motivation, reward-based learning, and mood-dependent decision-making, which are integral to cognitive engagement and performance. Cognitive deficits, therefore, often result from imbalances or dysfunctions in these systems, as seen in disorders like schizophrenia, ADHD, autism, and Alzheimer's disease. Importantly, cognition is not static. It is plastic and modifiable through environment. These behaviors are interpreted as evidence of higher-order cognition. They require evaluating alternatives, anticipating outcomes, and choosing optimal strategies. These processes are influenced by both rational computation and emotional modulation, the latter involving structures such as the amygdala and orbitofrontal cortex. Neuroeconomic studies decision-making and problem-solving represent the most advanced levels of cognition. They require evaluating alternatives, anticipating outcomes, and choosing optimal strategies. These processes are influenced by both rational computation and emotional modulation, the latter involving structures such as the amygdala and orbitofrontal cortex. Neuroeconomic studies reveal that even non-human primates and rodents exhibit rudimentary forms of decision-making, including risk assessment and delayed gratification, illustrating that the roots of cognitive processes extend deep into the animal kingdom. Social cognition, the capacity to perceive, interpret, and respond to the behaviors and intentions of others, is crucial in animals that live in complex societies. It involves theory of mind, empathy, and recognition of social hierarchies. Primates, elephants, dolphins, and certain birds such as corvids show impressive cognitive abilities such as mirror self-recognition, imitation, and tool use. These behaviors are interpreted as evidence of higher-order cognition, supported by evolutionarily expanded cortical regions and enriched life experiences. On the molecular and cellular level, cognition is underpinned by synaptic plasticity, neurotransmitter dynamics, and gene regulation. Glutamatergic signaling, especially via NMDA receptors, is central to synaptic changes involved in learning and problem-solving. Modulators like dopamine and serotonin influence motivation, reward-based learning, and mood-dependent decision-making, which are integral to cognitive engagement and performance. Cognitive deficits, therefore, often result from imbalances or dysfunctions in these systems, as seen in disorders like schizophrenia, ADHD, autism, and Alzheimer's disease. Importantly, cognition is not static. It is plastic and modifiable through environmental enrichment, experience, and training. Studies on neurogenesis in the adult hippocampus suggest that learning and cognitive challenges promote neuronal growth and integration, reinforcing the adaptive nature of cognition. This neuroplasticity forms the basis of cognitive therapies and rehabilitation strategies in clinical neuroscience. In summary, cognition integrates sensory perception, memory, emotion, attention, and decision-making through a network of brain regions, neural circuits, and molecular pathways. It is dynamic, context-dependent, and a product of both evolution and learning. For CSIRNet Life Sciences, understanding cognition provides crucial insight into how organisms perceive and interact with their world, a key theme linking neurobiology, behavior, and evolution. Previous year CSIRNet questions on cognition, CSIRNet Deck 2014, Part C. Which brain region is primarily associated with higher cognitive functions such as decision-making, planning, and attention regulation? A. Hippocampus. B. Amygdala. C. Prefrontal cortex. D. Cerebellum. Answer. C. Explanation. The prefrontal cortex is the seat of executive functions critical to cognition. CSIRNet June 2019, Part C. Which of the following statements about cognition in animals is true? A. Only primates show tool use and planning behavior. B. Cognition is exclusively dependent on language. C. Certain bird species exhibit cognitive traits like problem-solving and tool use. D. Cognition cannot evolve in non-mammalian species. Answer. C. Explanation. Birds like crows and parrots demonstrate problem-solving and tool use, indicating advanced cognition. CSIRNet Deck 2020, Part B. The neurotransmitter most involved in modulating reward-based decision-making and cognitive tasks is GABA. B. Dopamine. C. Serotonin. D. Acetylcholine. Answer. B. Explanation. Dopamine is crucial for reward prediction, motivation, and reinforcement learning. Let me know if you'd like the next lecture on biological clocks, sleep, emotion, learning models, or any other CSIRNet topic. I'll prepare it with the same quality and detail. You said, sleep and arousal, ChatGPT said. Here is your CSIRNet JRF Life Sciences full-length lecture on sleep and arousal, written with maximum detail, no tables, and tailored to the JRF level based on previous year question trends. It includes 2 to 3 solved previous year CSIRNet questions with year and answers, and is designed to serve as your exclusive and final coaching material to help you crack the exam. Sleep and arousal, CSIRNet JRF Life Sciences lecture. Sleep is a highly conserved physiological process observed across the animal kingdom, characterized by a reversible state of reduced responsiveness to external stimuli, decreased movement, and specific neurophysiological patterns. It is essential for survival, optimal brain function, memory consolidation, metabolic regulation, and synaptic homeostasis. The process of sleep is regulated by intricate interactions between circadian rhythms, homeostatic drive, and neurotransmitter-mediated arousal systems. Sleep is broadly classified into 2 main stages: non-rapid eye movement (NREM) sleep and rapid eye movement (REM) sleep. NREM sleep consists of multiple stages, progressing from light to deep sleep, marked by progressive synchronization of electroencephalogram (EEG) activity. In contrast, REM sleep is characterized by desynchronized EEG patterns resembling wakefulness, rapid eye movements, and muscle atonia. REM sleep is most closely associated with vivid dreaming and is crucial for emotional regulation and memory integration. The regulation of sleep involves complex neuroanatomical circuits. The process of sleep is regulated by intricate interactions between circadian rhythms, homeostatic drive, and neurotransmitter-mediated arousal systems. Sleep is broadly classified into 2 main stages: non-rapid eye movement (NREM) sleep and rapid eye movement (REM) sleep. NREM sleep consists of multiple stages, progressing from light to deep sleep, marked by progressive synchronization of electroencephalogram (EEG) activity. In contrast, REM sleep is characterized by desynchronized EEG patterns resembling wakefulness, rapid eye movements, and muscle atonia. REM sleep is most closely associated with vivid dreaming and is crucial for emotional regulation and memory integration. The regulation of sleep involves complex neuroanatomical circuits. The hypothalamus, specifically the ventrolateral preoptic nucleus (VLPO), plays a critical role in sleep initiation by inhibiting arousal-promoting centers through the release of inhibitory neurotransmitters such as GABA and galanin. The brainstem and basal forebrain contain wake-promoting centers such as the locus ceruleus, noradrenaline, raphe nuclei, serotonin, tuberomammillary nucleus, histamine, and cholinergic neurons, all of which interact with the thalamus and cortex to sustain arousal and attention. Arousal, in contrast, refers to the state of alertness or wakefulness, allowing organisms to respond actively to external stimuli. The reticular activating system (ROS) in the brainstem is pivotal for maintaining cortical activation and attention. Arousal levels can range from drowsiness to full alertness and are modulated by sensory inputs, emotional states, and circadian influences. The ascending arousal system is composed of multiple neurotransmitters, including dopamine, acetylcholine, norepinephrine, and orexin hypocretin, each contributing to different dimensions of alertness and cognitive engagement. One of the most crucial regulatory mechanisms in sleep-wake regulation is the circadian clock, located in the suprachiasmatic nucleus (SCN) of the hypothalamus. The SCN receives direct input from intrinsically photosensitive retinal ganglion cells (IPRGCs), allowing light to synchronize endogenous rhythms with environmental day-night cycles. Circadian regulation ensures a temporal alignment of sleep with the dark phase in diurnal animals. Disruption in this alignment, as seen in shift workers or jet lag, can impair sleep quality and lead to cognitive dysfunction, metabolic disturbances, and mood disorders. The homeostatic sleep drive is another major component, governed by the accumulation of sleep-promoting substances such as adenosine during wakefulness. Adenosine acts on A1 receptors in the basal forebrain to inhibit arousal-promoting neurons, facilitating the transition to sleep. Caffeine, a common stimulant, exerts its wake-promoting effects by blocking adenosine receptors. Sleep has profound effects on learning, memory, immune function, and metabolism. Slow-wave sleep (SWS) during NREM is essential for consolidating declarative memories, while REM sleep supports procedural memory, creativity, and emotional processing. Chronic sleep deprivation impairs cognitive performance, reduces alertness, and disrupts hormonal balance. In experimental studies, animals deprived of REM sleep showed increased irritability, anxiety-like behaviors, and even death after prolonged deprivation, emphasizing the essential nature of sleep. The flip-flop switch model describes how sleep and arousal states are stably maintained. In this model, VLPO neurons inhibit arousal centers and vice versa. Mutual inhibition between sleep-promoting and wake-promoting neurons ensures a rapid and stable transition between sleep and wake states. However, this system can become unstable in conditions such as narcolepsy, a neurological disorder characterized by excessive daytime sleepiness and sudden transitions into REM sleep. Narcolepsy is associated with a deficiency of orexin hypocretin, a neuropeptide produced by neurons in the lateral hypothalamus that stabilizes the sleep-wake switch. Slow-wave sleep (SWS) during NREM is essential for consolidating declarative memories, while REM sleep supports procedural memory, creativity, and emotional processing. Chronic sleep deprivation impairs cognitive performance, reduces alertness, and disrupts hormonal balance. In experimental studies, animals deprived of REM sleep showed increased irritability, anxiety-like behaviors, and even death after prolonged deprivation, emphasizing the essential nature of sleep. The flip-flop switch model describes how sleep and arousal states are stably maintained. In this model, VLPO neurons inhibit arousal centers and vice versa. Mutual inhibition between sleep-promoting and wake-promoting neurons ensures a rapid and stable transition between sleep and wake states. However, this system can become unstable in conditions such as narcolepsy, a neurological disorder characterized by excessive daytime sleepiness and sudden transitions into REM sleep. Narcolepsy is associated with a deficiency of orexin hypocretin, a neuropeptide produced by neurons in the lateral hypothalamus that stabilizes the sleep-wake switch. Narcolepsy is associated with a deficiency of orexin hypocretin, a neuropeptide produced by neurons in the lateral hypothalamus that stabilizes the sleep-wake switch. Another condition, insomnia, involves the inability to initiate or maintain sleep, often due to hyperarousal. It is commonly associated with stress, anxiety, or disrupted circadian rhythms. In contrast, REM sleep behavior disorder involves the loss of muscle atonia during REM, leading to dream and acting behaviors. These disorders underscore the complex balance of neural mechanisms that sustain sleep and arousal. From an evolutionary perspective, sleep may have developed to reduce energy expenditure, avoid predators during vulnerable periods, and facilitate brain plasticity. Comparative studies in animals such as fruit flies, Drosophila, zebrafish, and mammals demonstrate sleep-like states and reveal conserved genetic and molecular pathways, including period per and timeless TIM genes involved in circadian rhythm regulation. In summary, sleep and arousal represent dynamic, interdependent states governed by neural circuits, neurotransmitters, and genetic regulators. In summary, sleep and arousal represent dynamic, interdependent states governed by neural circuits, neurotransmitters, and genetic regulators. Understanding these processes provides crucial insight into how organisms manage energy, process information, and maintain internal physiological balance. For CSIRNet Life Sciences, a deep understanding of these mechanisms is vital for tackling questions that intersect neurobiology, behavior, and physiology. Previous year CSIRNet questions. Sleep and arousal. CSIRNet Deck 2017, Part C. Which of the following brain regions is primarily involved in the regulation of sleep-wake cycles? A. Hippocampus. B. Suprachiasmatic nucleus. SCN. C. Amygdala. D. Cerebellum. Answer. B. Explanation. The SCN is the master circadian pacemaker coordinating daily sleep-wake rhythms. CSIRNet June 2014, Part B. Which neurotransmitter is most closely associated with promoting wakefulness and is deficient in narcolepsy? A. GABA. B. Dopamine. C. Orexin. Hypocretin. D. Serotonin. Answer. C. Explanation. Orexin stabilizes the sleep-wake transition. Its loss causes narcolepsy. CSIRNet Deck 2016, Part C. REM sleep is characterized by which of the following features? A. Muscle rigidity and slow brain waves. B. Rapid eye movement, muscle atonia, and dream activity. C. Increased muscle tone and delta waves. D. Absence of EEG activity and eye movement. Answer. B. Explanation. REM sleep includes rapid eye movements, vivid dreams, and muscular atonia. Let me know if you'd like. Explanation. Orexin stabilizes the sleep-wake transition. Its loss causes narcolepsy. CSIRNet Deck 2016, Part C. REM sleep is characterized by which of the following features? A. Muscle rigidity and slow brain waves. B. Rapid eye movement, muscle atonia, and dream activity. C. Increased muscle tone and delta waves. D. Absence of EEG activity and eye movement. Answer. B. Explanation. REM sleep includes rapid eye movements, vivid dreams, and muscular atonia. Let me know if you'd like the next lecture on biological clocks, emotion, learning theories, or any other topic from the CSIRNet syllabus. I'll make it just as complete and exam-oriented. You said, biological clocks. ChatGPT said. Here is your CSIRNet JRF Life Sciences full-length lecture on biological clocks, written in maximum detail, paragraph format with no tables, and tailored to the JRF level. It focuses on previously asked question areas and includes 2 to 3 CSIRNet previous year questions with year and answers. This is designed as your final, exclusive coaching material, helping you master this topic thoroughly for the exam. Biological clocks. CSIRNet Life Sciences lecture. Biological clocks are internal time-keeping mechanisms that allow organisms to regulate physiological, biochemical, and behavioral processes in synchrony with predictable environmental cycles such as the day-night rhythm, lunar cycle, tidal changes, or seasonal variations. These clocks enable organisms to anticipate changes in the environment rather than merely reacting to them, thereby conferring a significant evolutionary advantage. The most well-studied biological rhythm is the circadian rhythm, which follows an approximately 24-hour cycle. Circadian rhythms regulate daily fluctuations in sleep-wake cycles, hormone secretion, body temperature, feeding behavior, and gene expression. The suprachiasmatic nucleus (SCN) of the hypothalamus is the principal pacemaker in mammals. Located above the optic chiasm, the SCN contains a set of neurons that generate rhythmic patterns of activity via transcriptional translational feedback loop (TTFL). These neurons maintain rhythmic expression of clock genes, which include clock, BMAL1, PER period, and cryptochrome. Clock and BMAL1 heterodimerize and promote the transcription of PER and CRY. As PER and cry proteins accumulate in the cytoplasm, they form complexes that translocate to the nucleus and inhibit the activity of clock BMAL1, thereby repressing their own transcription. This creates approximately 24-hour oscillation in gene expression. In addition to the central clock in the SCN, peripheral clocks exist in other tissues such as the liver, lungs, kidney, and even individual cells. These peripheral clocks are also based on the same molecular feedback loop but are entrained by systemic signals such as feeding, body temperature, and hormones like glucocorticoids. The SCN synchronizes these peripheral clocks, ensuring organism-wide temporal coordination. Photoreception and light entrainment of the circadian clock involve a specialized set of retinal ganglion cells expressing melanopsin, which are intrinsically photosensitive and distinct from rods and cones. These cells project directly to the SCN via the retinohypothalamic tract, allowing light information to reset the circadian rhythm. This is particularly important for entraining the sleep-wake cycle to the external light. This is particularly important for entraining the sleep-wake cycle to the external light-dark cycle. The output of the SCN regulates the pineal gland, which secretes the hormone melatonin during the dark phase of the light cycle. Melatonin feeds back to the SCN and other tissues, acting as a hormonal signal for darkness and helping regulate sleep timing, seasonal rhythms, and reproductive cycles in photoperiod-sensitive organisms. Ultradian rhythms are cycles shorter than 24 hours, e.g., the basic rest activity cycle or hormone secretion cycles, whereas infraradian rhythms exceed 24 hours, e.g., menstrual cycle or seasonal breeding. All these biological rhythms can be governed by similar oscillator mechanisms, although the circadian clock is the best characterized. Melatonin feeds back to the SCN and other tissues, acting as a hormonal signal for darkness and helping regulate sleep timing, seasonal rhythms, and reproductive cycles in photoperiod-sensitive organisms. Ultradian rhythms are cycles shorter than 24 hours, e.g., the basic rest activity cycle or hormone secretion cycles, whereas infraradian rhythms exceed 24 hours, e.g., menstrual cycle or seasonal breeding. All these biological rhythms can be governed by similar oscillator mechanisms, although the circadian clock is the best characterized. In model organisms like Drosophila melanogaster, the genetic and molecular basis of biological clocks has been extensively dissected. In Drosophila, per and TIM are the core feedback repressors, and their expression oscillates over the course of the day. Mutations in period per and timeless TIM genes cause changes in the free-running period or even abolish rhythmicity altogether. These discoveries led to the awarding of the 2017 Nobel Prize in Physiology or Medicine for the elucidation of molecular mechanisms controlling circadian rhythms. Disruptions in biological clocks can have profound health consequences. Circadian misalignment, such as that seen in shift workers or individuals with jet lag, leads to impaired cognition, metabolic dysfunction, depression, and increased risk of chronic diseases. The circadian clock also affects drug metabolism, and thus the field of chronic pharmacology explores how the timing of drug administration can influence therapeutic efficacy and toxicity. Studies in rodents and humans have shown that lesions to the SCN abolish circadian rhythms of activity, hormone secretion, and body temperature. Transplantation of SCN tissue can restore rhythmicity, demonstrating the autonomy and hierarchical control of the central clock. Even in plants and cyanobacteria, circadian rhythms are generated by transcriptional or post-translational feedback mechanisms. In cyanobacteria, the Chi-ABC system produces a self-sustained rhythm in phosphorylation status of proteins, independent of gene transcription. From an evolutionary perspective, biological clocks enhance fitness by optimizing the timing of key activities such as feeding, reproduction, and predator avoidance. For example, nocturnal and diurnal species exhibit contrasting circadian phase relationships with respect to melatonin secretion and core body temperature rhythms, illustrating the adaptability of internal clocks to ecological niches. In conclusion, biological clocks are ubiquitous and tightly regulated systems that orchestrate the timing of physiological and behavioral processes. Understanding their molecular basis, neural circuitry, and physiological outputs is vital for decoding how organisms maintain homeostasis in a rhythmic environment. For CSIRNet Life Sciences, mastering this topic means not only grasping the molecular feedback loops and brain regions involved but also their ecological, physiological, and pathological implications. Previous year CSIRNet questions. Biological clocks. CSIRNet Deck 2015. Part B. Which of the following brain regions acts as the master circadian pacemaker in mammals? A. Hippocampus. B. Suprachiasmatic nucleus. SCN. C. Amygdala. D. Prefrontal cortex. Answer. B. Explanation. The SCN in the hypothalamus orchestrates circadian rhythms across the body. CSIRNet June 2016. Part C. Mutations in the per and TIM genes in Drosophila disrupt which biological process? A. Wing development. B. Reproductive behavior. C. Circadian rhythm regulation. D. Chemosensory signaling. Answer. C. Explanation. The per and TIM genes encode core components of the fly circadian clock. CSIRNet Deck 2017. Part B. Which hormone, secreted in darkness, is regulated by the SCN and involved in circadian rhythm modulation? A. Cortisol. B. Insulin. C. Melatonin. D. Ghrelin. Answer. C. Explanation. Melatonin is secreted by the pineal gland during darkness under SCN control. Let me know if you'd like the next lecture on learning theories, emotion, communication, or any topic in CSIRNet Life Sciences. I'll make it with the same level of detail, depth, and exam focus. You said, development of behavior, ChatGPT said. Here is your CSIRNet JRF Life Sciences full-length lecture on development of behavior, prepared at the highest standard, from base to advanced, with maximum word limit, paragraph-only format, no tables, and based on previous year CSIRNet question areas. It includes 3 previous year questions with answers and years mentioned, as you requested. This is billed as your only coaching material, with nothing left out. Development of behavior, CSIRNet Life Sciences lecture. The development of behavior, or ontogeny of behavior, refers to the process by which behavioral traits emerge and become refined throughout the lifespan of an organism, beginning from embryogenesis to adulthood. It involves a complex interplay of genetic, epigenetic, neurobiological, environmental, hormonal, and social factors, all contributing to the shaping of both instinctive and learned behaviors. Behavioral development is not a fixed or predetermined process. It is a dynamic system that is sensitive to critical periods, external stimuli, and internal physiological states. Behavioral development begins in the early embryonic stages, where gene expression patterns in the nervous system lay down the framework for future responses. Genes provide the blueprint for neural circuits responsible for specific behaviors. However, these circuits are modifiable and often require postnatal experiences for fine-tuning. For example, in birds, the song learning behavior is genetically programmed but requires auditory experience and social interaction for proper song acquisition, demonstrating the importance of interaction between genetic predisposition and environmental input. A classic illustration of behavioral development is seen in imprinting, a phenomenon first described by Konrad Lorenz. Imprinting occurs during a critical or sensitive period shortly after birth when young animals, such as ducklings, form strong associations with the first moving object they see, usually their mother. This form of learning is rapid, irreversible, and affects future social and reproductive behaviors. Imprinting exemplifies how developmental timing and exposure to specific stimuli are crucial for certain behaviors to emerge correctly. Hormonal regulation plays a key role in shaping behavior during development. For example, in rodents and humans, exposure to sex steroids like testosterone or estradiol during specific periods of brain development can influence sexually dimorphic behaviors. The differentiation of male and female behaviors, such as aggression or parenting style, often results from such organizational effects of hormones, followed later by activational effects during puberty or adulthood. Disruptions in the hormonal environment during development, such as by endocrine disruptors, can lead to aberrant behaviors. Neurodevelopment and synaptic plasticity also underpin behavioral maturation. The formation of neural connections during critical periods determines the capacity for sensory perception, motor coordination, and learning. For instance, in mammals, visual and auditory inputs during early life are essential for the normal development of vision and hearing. Deprivation of such inputs during sensitive windows, e.g., occluding one eye in kittens, can result in permanent deficits, showing that experience expected plasticity is vital for normal behavior. Learning mechanisms, such as habituation, sensitization, classical conditioning, and operant conditioning, begin to function early in life and are key contributors to behavioral development. In precocial species, those that are relatively mature at birth, such learning starts before or shortly after birth. In ultrashort species, those born in an undeveloped state, learning continues over an extended period as the brain matures postnatally. These differences highlight the role of species-specific life history strategies in shaping behavioral development. Social environment and parental care significantly affect behavioral outcomes. In primates, including humans, maternal behaviors such as grooming, nursing, and protection are critical for the infant's emotional and cognitive development. Deprivation of maternal care or exposure to abusive interactions can lead to long-term alterations in stress reactivity, sociability, and anxiety-related behaviors. Studies on Harlow's monkeys show that infant rhesus monkeys raised without maternal contact exhibited abnormal social and sexual behaviors, proving that early social interaction is essential for normal behavioral development. Epigenetic modifications also play a key role in the development of behavior. Environmental factors such as maternal care, nutrition, and stress can lead to DNA methylation or histone modifications that alter gene expression without changing the underlying DNA sequence. These modifications can have lasting effects on behavior, including anxiety, fear responses, and learning ability. Epigenetic regulation links early life experiences with persistent behavioral phenotypes and offers a mechanism by which nurture influence. Epigenetic modifications also play a key role in the development of behavior. Environmental factors such as maternal care, nutrition, and stress can lead to DNA methylation or histone modifications that alter gene expression without changing the underlying DNA sequence. These modifications can have lasting effects on behavior, including anxiety, fear responses, and learning ability. Epigenetic regulation links early life experiences with persistent behavioral phenotypes and offers a mechanism by which nurture influences nature. Experience-dependent plasticity ensures that the brain continues to adapt to environmental inputs throughout life. Behavioral flexibility and adaptation are maintained through synaptic remodeling, neurogenesis in certain brain regions, e.g., hippocampus, and changes in neurotransmitter systems. However, critical periods represent windows during which experience has a uniquely strong effect. After these periods close, some forms of learning, like language acquisition in humans, become much more difficult. From an evolutionary perspective, developmental plasticity in behavior allows organisms to fine-tune their responses to match local environmental conditions, improving survival and reproduction. The reaction norm of behavior, the range of behaviors an individual can display depending on the environment, results from the complex interactions between genes and experiences. Therefore, studying the development of behavior offers insights not only into individual variability but also into the adaptability and evolution of behavioral traits. In experimental contexts, model organisms like zebrafish, Drosophila, and rodents have been used extensively to study the genetic and neural basis of behavioral development. For instance, knockout studies in mice have revealed the role of specific genes in maternal behavior, aggression, and social preference. Transgenic lines in Drosophila help in tracing the development of neural circuits underlying courtship behavior. These studies demonstrate how molecular genetics can unravel the developmental basis of complex behaviors. In summary, the development of behavior is a multifaceted process governed by genetic programming, neural circuitry, hormonal signaling, learning, environmental inputs, and social context. This developmental trajectory is modulated by critical periods, plasticity, and feedback from behavior itself. Understanding these mechanisms provides crucial insights into how behavioral traits are formed, maintained, and modified, which is essential for cracking the CSIR-NET JRF Life Sciences exam, especially in the behavioral biology unit. CSIR-NET previous year questions. Development of behavior. CSIR-NET Deck 2014, Part C. Imprinting in birds is an example of which of the following? A classical conditioning. B. Operant learning. C. Learning during a sensitive period. D. Observational learning. Answer. C. Explanation. Imprinting is a form of learning that occurs only during a sensitive period early in life. CSIR-NET June 2018, Part B. Harlow's experiments with infant monkeys reveal the importance of which factor in behavioral development? A. Food availability. B. Parental grooming. C. Maternal contact comfort. D. Vocal learning. Answer. C. Explanation. Monkeys preferred soft surrogate mothers, showing the importance of contact comfort. CSIR-NET Deck 2016, Part C. Which of the following best explains how early life experiences can result in long-term changes in behavior without altering DNA sequence? A. Genetic mutations. B. Epigenetic modification. C. Neurogenesis. D. Protein phosphorylation. Answer. B. Explanation. Epigenetic changes like methylation mediate long-term behavioral changes from early experiences. Would you like the next lecture on learning and memory, emotion, communication, or evolution of behavior? I'll make it in the same exam-focused, comprehensive format. You said. Social communication. ChatGPT said. You said. Social communication. ChatGPT said. Here is your CSIR-NET JRF Life Sciences full-length lecture on social communication, crafted at the highest possible quality, covering the topic from base to advanced level, with no tables, maximum word limit, and paragraph-only format. It is based on previous year CSIR-NET questions, and includes pre-solved previous year questions with answers in years mentioned. This is your exclusive study material, leaving nothing important out. Social communication, CSIR-NET Life Sciences lecture. Social communication is the exchange of information between individuals of the same species, which serves to coordinate behavior, establish social bonds, mediate conflict, and facilitate reproduction. In the context of animal behavior, communication is not just about vocal sounds but encompasses a wide variety of signals that include visual cues, chemical signals, tactile contact, vibrational and auditory signals, and even electrical impulses in some taxa. These signals are evolutionary adaptations, fine-tuned to function efficiently in specific ecological and social contexts. A signal is any act or structure produced by a sender that influences the behavior of a receiver, and which has evolved specifically for that purpose. Unlike cues, which may incidentally provide information, signals are shaped by natural selection because they benefit either the sender, the receiver, or both. Social communication is often mutualistic, but not always. Manipulative communication, as in deceptive signals, can also evolve if it increases the sender's fitness. Social communication thus has profound implications for survival, reproductive success, and social structure. In visual communication, animals use body coloration, posture, facial expressions, and bioluminescence to convey information. For example, the colorful plumage of peacocks, or the threat displays of lizards, convey dominance, readiness to mate, or aggression. Visual signals are especially useful in daylight environments with open habitats, but are limited in low visibility conditions. Auditory signals are widely used, especially in birds, mammals, amphibians, and insects. They allow communication over long distances in intense environments such as forests. Examples include territorial songs of birds, alarm calls in primates, and mating calls in frogs. These signals can be rapidly modulated and can carry complex information. In many species, vocal learning is involved in acquiring and refining calls, such as in songbirds and cetaceans. The context specificity and variation in pitch, frequency, and duration make auditory signals highly flexible. Chemical communication, or pheromonal signaling, is used extensively in insects and mammals. Pheromones are chemicals secreted externally that elicit specific behavioral or physiological responses in conspecifics. In ants, pheromones are used for trail marking, alarm signaling, and colony cohesion. In mammals, pheromones play a role in mate attraction, territory marking, and mother-offspring recognition. Chemical signals are especially effective in dark environments, are long-lasting, and can persist even after the sender has left the area. However, they are less directional and slower than visual or auditory signals. Tactile communication involves physical contact and is especially significant in animals with close social bonds. Grooming in primates is a key example, reinforcing social structure and alliances. In honeybees, the famous waggle dance combines tactile and vibrational cues to communicate the direction and distance of food sources. Tactile signals are also used in courtship, parental care, and dominance displays. Electrical communication occurs in some aquatic species, like electric fish, which generate and detect weak electric fields to identify conspecifics, navigate, and detect prey. It is particularly useful in murky waters where visual signals are ineffective. The evolution of communication involves signal design shaped by the environment, sensory drive hypothesis, receiver psychology, and the costs and benefits to both sender and receiver. Honest signals are maintained through handicap principles, e.g., elaborate traits that are costly to fake, while deceptive communication also exists, such as false alarm calls used to distract rivals. For instance, some birds give alarm calls when competing for food to cause others to flee, gaining an opportunity to monopolize the resource. Social communication is essential for cooperative behavior, including coordinated hunting, group defense, alarm signaling, parental care, and group cohesion. In eusocial insects like bees and ants, complex communication systems regulate division of labor and colony organization. In social mammals, like wolves and elephants, vocalizations, body language, and scent marking play crucial roles in maintaining social hierarchies and reproductive roles. An interesting example of symbolic and referential communication is seen in vervet monkeys, who produce distinct alarm calls for different predators, e.g., leopards, eagles, snakes, and group members respond with appropriate evasive behaviors. This indicates that animal communication can convey specific information, not just general arousal. In primates and humans, communication becomes increasingly complex, with evidence for intentionality, syntax, and semantic content in some species. Studies in great apes and dolphins suggest a capacity for referential signaling, and in human evolution, language emerges as an extreme refinement of social communication, enabling the formation of large, stable social groups and cultural transmission. The neural basis of communication involves specialized brain circuits for producing and interpreting signals. For example, in songbirds, regions such as the HVC and RA nucleus control song production, and lesions in these areas disrupt learned vocalizations. Similarly, in mammals, limbic structures, auditory cortex, and olfactory bulbs process communication signals critical for social behavior. In summary, social communication is a cornerstone of animal behavior, supporting cooperation, competition, reproduction, and survival. It is shaped by natural selection to match sensory modalities with ecological demands and social structures. For the CSIR-NET exam, understanding the modalities, evolution, functions, and examples of communication across taxa is crucial, along with their neural and hormonal correlates. CSIR-NET previous year questions. Social communication. CSIR-NET June 2016, Part B. Which of the following is an example of referential communication in animals? A courtship display in peacocks. B. Alarm calling in vervet monkeys. C. Trail pheromones in ants. D. Grooming in primates. Answer B. Explanation. Vervet monkeys use different alarm calls for different predators, conveying specific information. CSIR-NET Deck 2015, Part C. In honeybees, the waggle dance communicates which of the following information? A location of the queen. B. Direction and distance of food. C. Imminent danger. D. Egg laying site. Answer B. Explanation. The waggle dance conveys both direction and distance of the food source from the hive. CSIR Net June 2020, Part B. What is the primary advantage of chemical communication in ants? A. It is faster than visual communication. B. It is not energetically costly. C. It persists after the sender is gone. D. It is species-specific only in mammals. Answer. C. Explanation. Chemical trails and pheromones persist in the environment, allowing others to follow them later. Let me know when you're ready for the next lecture. Would you like dominance hierarchies, territoriality, or reproductive success in mating systems? Next. I'll prepare it in the same ultra-detailed format. You said. Social dominance. ChatGPT said. Here is your CSIR Net JRF Life Sciences ultra-detailed lecture on social dominance, written at the JRF level, in paragraph-only format without any tables, with maximum word limit, and based on previous CSIR Net question areas. This is designed to be your only coaching material, covering basic to advanced concepts and including 3 previous year questions with answers in year mentioned. Social dominance. CSIR Net Life Sciences lecture. Social dominance refers to the hierarchical organization of individuals within a group based on access to resources such as food, mates, and space. These dominance relationships are usually established and maintained through direct interactions like aggressive encounters, displays, threats, and submissive behaviors. Over time, stable hierarchies emerge in many social animals, reducing the frequency of actual conflicts and establishing predictable patterns of behavior. Dominance is not just about aggression; it reflects a socially recognized rank that is reinforced through repeated interactions and social signals. Dominance hierarchies can be linear or nonlinear, stable or dynamic, and may differ between sexes, age classes, or seasonal contexts. In a linear hierarchy, also called a pecking order, each individual dominates those ranked below and submits to those ranked above. For example, in chickens and wolves, linear dominance orders are common, leading to efficient resource allocation without constant fighting. In nonlinear hierarchies, dominance relationships are more complex and may involve triangular or looped interactions, where A dominates B, B dominates C, but C may dominate. These are more common in species with complex social systems or changing group compositions. Dominance can be context dependent, where an individual may be dominant in one context, e.g., feeding, but subordinate in another, e.g., mating. It may also be absolute or relative. A dominant individual in one group may not be dominant in another. The factors influencing dominance include body size, strength, age, experience, prior victories, winner effect, hormonal state, especially testosterone levels, and even social alliances. In primates, for example, individuals often gain and maintain high rank through coalitions and alliances, not just brute strength. The advantages of dominance are significant. Dominant individuals typically enjoy greater access to food, preferred mating partners, safer resting sites, and higher reproductive success. In many species, dominant males father more offspring, while dominant females may have priority access to food, enhancing their reproductive output. However, dominance also comes with costs, such as higher energy expenditure in maintaining status, greater risk of injury during challenges, and elevated stress levels, particularly in unstable hierarchies. In contrast, subordinate individuals may adopt alternative strategies to survive and reproduce. These include sneaky mating tactics, delayed reproduction, or emigration. Some species exhibit conditional strategies, where individuals switch roles based on environmental or social cues. For example, in some fish species, smaller males mimic females to sneak fertilizations without engaging in direct competition with dominance. Dominance hierarchies may be maintained through ritualized displays and signals that avoid costly fights. These include threat gestures, postural displays, facial expressions, and vocalizations. For example, baboons display threatening yawns showing canines, while wolves use tail posture and eye contact to assert rank. Submission signals, like grooming, crouching, or avoiding eye contact, serve to reinforce the social order without escalating conflict. In primate societies, social dominance is often closely tied to coalitionary behavior. Males and females form alliances to defend or contest status. High-ranking individuals often use social bonds, grooming networks, and political strategies to maintain their rank. For instance, in chimpanzees, males that form long-term alliances are more successful in attaining and holding dominant positions. Dominance also influences stress physiology. Subordinates in unstable hierarchies often have elevated glucocorticoid levels, which can impair health and reproduction. Some species exhibit sex-specific hierarchies, where males and females maintain separate dominance ranks. Immature.